Before I go into today's lessons, couple of questions. I did go live on YouTube last Monday. Did anybody watch it? You did? How was it?
I was there from New York.
You could
From New York to login. That's
you could make comments, right?
Yeah.
Okay.
Yeah, part of it froze, like the bunny
Yeah, at
part.
the end,
The
there's
bunny part.
yeah
It was
the
just, I
cortex
skipped
of bunnies, and it just
Yeah.
froze there.
Yeah, it's
The
froze.
screen, at least.
I thought it
You
was
still
mine.
hear the voices.
Oh,
Yeah.
You
really?
can still hear your voice, but you could, but you were just looking at that one slide. Yeah, like for 20 minutes.
Oh, really?
Yeah.
Okay, so it didn't really work out that well.
No, the first part was fine.
No,
The
it's
second part
good at
got
it.
stuck.
Except something
Okay,
froze, bunny.
Second thing I wanted to
Final.
tell you, there is a, so we eventually came up with this course reader. It's called Heredity Reader. Did anybody take a look at this? You did?
Kind of.
Okay, kind of. Okay, I just want to, this is basically taken from a biology book, and it pretty much covers everything that we have gone through. So what, and the assignments are now going to come from this particular book. So they'll say page number so and so, these problems. That'll be like the first set of assignments that will come. So the plan right now is by the 17th of December, all the classes will be done, all the topics will be covered. Okay? And then after the Christmas break, really what you'll work on are the assignments. And we'll still meet weekly in the class to go over the assignments and answer questions. But these lessons are really going to stop after December 17th because we should be covering all the material. So what the plan is that, and you can see all the stuff that has been covered is actually here, and you can read about it. And okay, any questions? Okay. Second thing is there is also this course syllabus. This course syllabus actually, so the assignment coach means we have divided among the coaches the different topics on which there will be assignments. So these are the coaches who will provide the assignment. I will also fill up the section here which says which page number or which section in the reader that I just showed. If you want to know about this, it's in this video lesson, and this is the place in the reader that you will have it. Yes?
Where are we right now?
Right now, we are going to talk about human sex determination, sex-linked traits, pedigree, construction, analysis of karyotypes, abnormal production of gametes with abnormal
Number.
chromosomes, and addition and translocation. This is what will be covered in the next 2 classes, and this as well. And then the transcription, translation, gene-protein relationship, DNA structure will be covered on the last day. And that will be done by December 17th by Professor Simpson. Right?
He's my
Yeah, he's
dad.
dad.
That's why I was smiling.
Good. Okay. Any other questions? Okay, and finally, these are some Science Olympiad notes that I got from the Science Ollie website, and this has sort of all the topics kind of compressed in these note form. So I thought this was useful for you to kind of keep as a short reference.
Which one is this again?
This is called the Sai Ollie Notes Heredity. Okay?
And I think they have to make their own sheet also, so
Yeah, I think
they have to meet individually. as
in 3 pages you have to make your final notes. So I think January we will really focus more on doing problems and really doing practice. And that's when I mean you'll have to go over the assignments. Okay? Okay, then let's go over some of the stuff. Now, basically, I have, till now what I have covered is the Mendel's laws, exceptions to Mendel's laws, meiosis and mitosis. And this is the foundation on which everything else is based here. And you can almost, if you followed those things, you should be able to understand why this is happening. So, I'll start with chromosomes. Okay? So, till now we have talked in the abstract about heredity. We said that, I mean, we know that things are transmitted from one generation to another, but what is the physical basis of it? And the answer lies in meiosis and mitosis. Because if you think about it, the life of any organism starts when 2 gametes fuse together and form this one cell, right? All life starts with this one cell. Does that make sense? So, whatever you are, all your phenotypes are determined by something that is there in that cell, right? Physically, there must be some substance. And initially people didn't really know what it was, because you saw the cell, right? There are many parts in the cell. Which specific part is the one that's keeping all this information and passing it from one generation to another is not obvious. And so there were a set of experiments that were done on fruit flies in the early part of last century, 1910 to 1930, by a person called Morgan. And he actually proved through his experiments that there were a bunch of other experiments which sort of showed that chromosomes in the cell were the actual carriers, or they carried the information of all your genotype and phenotype from one generation to another. And if you think about it, during meiosis, it was the chromosomes that separated, right? And so the chromosomes contain all the information that defines you as an individual. And if you pause a moment and think about this is actually a pretty deep thing. If you think about yourself, all of you as individuals, you have so many traits, right? Your nose, you have so many body parts, so many different attributes, probably in the tens of thousands. And all that is contained in a single cell which you can't even see with your naked eye, It is, and the chromosomes are even smaller than that, right? I mean, if you blow up by a factor of probably 10,000, you'll probably see these chromosomes under a microscope. So all this information is there in the chromosome. So what are chromosomes? So for example, humans have 23 pairs of chromosome, and one chromosome comes from each parent. Okay. So 23 comes from your father, 23 comes from your mother. This is how it works. And the reason for this is meiosis, right? Meiosis was the process of reduction. And the chromosomes are basically comprised of coiled up DNA, and we'll go into the atomic structure of DNA later, but for now you can think of this as one giant molecule which is called DNA. And unless there is any recombination during meiosis, all these chromosomes are just coming intact. So your father has 46 chromosomes, you choose 23 out of those randomly, 23 pairs. So you don't just choose 23 randomly, you choose one out of the two of the pair. Choose them randomly, same from your mother, and then fuse it together, get back the 46. That's the way it works. But if there is no recombination, then you just randomly get 23 out of the 46, right? What happens during recombination? Does anybody remember?
Like the 23rd pair, it
So-
decides if it's a male or female baby.
No.
It's
No,
the last one.
no. That's sex determination. Debangi, do you remember? Okay, Shohadra.
From the male side, one of the chromosomes may switch with the female side, like.
That's, when does recombination happen? During which phase?
It's in
Meiosis, right?
Yeah.
So in meiosis, there is no male or female. Meiosis happens independently in males
Oh,
and females,
formation, yeah.
right?
Yeah.
So when the gametes are formed, you have these.
When they split apart.
When they split apart.
Then some of the switch.
Yeah.
Some of the switch.
Yes.
So when they
Lower
split
side.
apart,
The
yeah.
higher.
Yes. And let me actually, instead of drawing on the board,
Should be on
let me
this
go to
side.
your reader and show you. There's a place in the reader where this is shown quite nicely.
Excuse me?
Yeah.
What do you mean by under normal conditions, all of the chromosomes are inherited intact?
That means it's not
That
split
means if there
like
is no
this
recombination,
recombination.
then the chromosomes just don't change, right?
Uh-huh.
But if there is recombination, something else happens, right? So, and if you remember, let's go to meiosis and hopefully this will be a good refresher. Okay, let me go to the meiosis slides. Mitosis and meiosis. And here, if you see, right, this is how they line up. And in recombination, what happens is a part of
This
a
green.
part of this chromatid and this chromatid swap over. See, you remember that? Sorry? And you can see that it's happened here. This is recombination. You see? This part has gone over there and this part has gone over there. Which now leads to a chromosome that is different from the original chromosome that you had.
Mhm.
But suppose this had not happened, you would either get this, either the red copy or the
Copy.
green copy, right? And that's what I meant under normal circumstances, which is okay. But recombination is completely normal. There is, in fact, during meiosis, probably 20 to 30 recombinations happen. So it is completely normal, and it does not lead to any abnormality because of that.
But the same parts always switch, right?
Yes.
The same sections.
Yes, and which is why what happens is you get another allele, but it's still the same gene, because, and that you'll see here. So chromosomal theory of inheritance is, it's saying that chromosomes contain the units of heredity, and these units of heredity are called genes. Physically, they are really segments in the chromosomes. Okay? And during meiosis, chromosome pairs segregate. Segregate means they separate, and each of the gamete or a sex cell has half the number of chromosomes as compared to a normal cell, right? And the chromosomes assort independently during meiosis, which means it is random which chromosome lands up in which gamete, and each chromosome contains many genes, right? This is now in chromosomes during meiosis, recombination and segregation are completely normal, but other things can happen, and these could change the structure of the chromosomes, and usually these are not good at all. They lead to all kinds of problems, and things that can happen are mutations, and in mutations what happens is the allele that you had just changes. There is a random change in the structure of the DNA, and you have a different allele. And then there are other things like deletion, duplication, inversion, and translocation, and we'll go over each of these that can happen. And usually, all of these lead to problems. So just like we had Mendel's laws, and then we had a bunch of exceptions, these are like exceptions to meiosis, the normal process of meiosis. Okay?
Wait.
Any questions? Okay. And then we have what are called sex chromosomes. And why are they called sex chromosomes? Because,
They determine the gender.
yes, they determine the sex or the gender of the individual. And so, the most of the chromosomes, all but one of the chromosome pairs, are called autosomatic, which means they are identical. So if you take a male or a female and you look at their chromosomes, you'll find 22 pairs are identical. Now, when you say identical, it doesn't mean that they are exactly the same, but they have the same genes in them. Okay? Different alleles, but same genes. Okay? And then we have the sex chromosomes, which are different between a male and a female. Okay? Sex linkage is really, if you have a gene which is located in your sex chromosome, then the phenotype that will result from that gene is going to be based on your gender. Does this make sense to you? No?
Repeat that?
So, if you think about it, all chromosomes have some genes in them, right? And each gene or a collection of genes is responsible for some characteristic that you have. That's basic, you know. Now, suppose that gene is present in your sex chromosome. What will happen? Yes.
It'll be carried by that sex chromosome. So if it's like if the person is if there's a disease that's like the female has and this is sex linked traits, right? So if it's linked to the female, if like the X from the XY, if it's linked to the X, then and there's another one that's also and it's dominant, and then this one does not have it, so it's and the girl does not have it, and it's and then and if it's a girl, then it would be have the disease, and if it's a guy, if it's a male, then it would not.
Okay, that's sort of, I mean, yeah, I think you're telling the right thing, but I'll just put it in a, okay, you have a different opinion about this?
It's it's just pretty much the same thing.
Almost same thing.
Yeah.
So I'll just rephrase it in a way which is a little bit more general. It doesn't have to be a disease. It says that if you have a gene which is located in your sex chromosome, I mean, let's say it's located in your Y chromosome, okay? A female will not even have it. A female doesn't have a Y chromosome. So depending on your sex or your gender, this phenotype will show up differently between males and females. That's really why these are called sex linkage. It's linked to your gender or your sex. And the reason why it is like this is because a male and a female have different chromosomes which determines their sex. Okay? Make sense? So here, for example, you have so females have these two X chromosomes, and suppose you have a particular gene in its, this is the dominant form, capital A, and this is the recessive form. So, and then you have this other combination. So you can have all these three different combinations of this particular gene in a female, and then in a male you have something different. You don't have another X, you have a Y, right? And what happens is, for example, in a female, even though this is recessive, right? XA is recessive. X small a is recessive. So whatever that phenotype is not going to show up for the female. Now just think about it. If you happen to be a male and you got this X small a, this is recessive, right? But Y doesn't even have that gene for small a. And what will happen is, even though it's recessive, it will show up because Y doesn't have it. Which means that
you can basically assume that
Yes, yes.
it does.
Because Y is a much smaller chromosome, and even if there is a recessive character in the X, in males it will show up, whereas in females it will not show up unless it is homozygous. So here, for example, a heterozygous
Yeah, it's the same.
yeah, heterozygote is going to show up this this A character, and this homozygous will show up this small a character. But in males, even though you have a recessive version of it, it will still show up. Okay, because there is nothing to nothing in Y to sort of counter it. So usually males are more susceptible to actually not usually, they are more susceptible to X-linked traits. X-linked trait means that gene is present in X. Now, if a gene is present in Y, and there are very few genes in Y, it's not going to affect a female. Okay.
Male is the only ones that have it.
Yeah. So, for example, take the case of the eye color in Drosophila. This is one of the flies that Morgan studied in his lab in the early 1900s. So here you have this is red. Red is dominant, and this little r is recessive, and recessive produces white eyes, and dominant produces red eyes. Okay, that's the. And unlike what Mendel studied, in this particular case, this gene happened to be in the sex chromosome, not in the normal autosomal chromosomes. Okay? So, this particular combination, this you should know right now. This produces red-eyed females, and this produces white-eyed female. And in case of males, the Y doesn't really do anything to it. Okay, so even though there is only one version of the recessive gene,
Yeah, there's nothing to counter it.
there's nothing to counter it. So this produces a white-eyed male. This still produces a red-eyed male because it's dominant. But this was not supposed to happen, right? Unless there was another X small r. So and now you can see what happens when you mate red-eyed females with white males, and this is like going to Mendel. This is like a purebred red-eyed female with a white-eyed male, and then you can do this. You'll see that in the first generation, all the females and all the males are red, right? And then in the next generation, you draw the Punnett square, and you'll find that all females have red eyes, half the males are white-eyed, and half the males are red eyes. And this is different from what we learned in Mendel, remember? In Mendel, it used to be like a 3:1 ratio. It had nothing to do with male or female. But now,
But why is it white? I don't understand that.
why is it white?
Because why is some of the males white? Because in the other one, the Y you said that it doesn't counter it. So why does
Because just do the Punnett square here, right? So you in the first generation you'll get all red females this way and all red males. But now when you do the segregation and you look at the combinations, you have this combination, right? This is So
Yeah.
these are the males, these are the females,
Uh-huh.
right? And here you have half of them, half of the males happen to have this X small r, and that is a white-eyed, right?
Yeah, but there's but still that's a recessive trait of that one. Then there's Y doesn't have counter it, so will it be white-eyed?
Because the recessive trait is
White.
white, it's
White.
coding
Oh,
for white.
Nothing's
Yeah.
countering it.
Okay.
Nothing is countering it.
Okay, this is-
Red
Yeah.
dominant.
Oh, so the lowercase r is coding for okay.
Yes,
Yeah.
yes,
So it's white.
Okay.
yes. And then you can see this other example also, which is white female with red males, and you can see all kinds of interesting things happen, right? And it's still applying the same principles that you learned from Mendel, but the results are different. And the reason why they are different is males and females, if you have a gene in the sex chromosome, things become different. Okay?
Mm.
Any questions about that? Okay, good. So, here are some examples of sex-linked traits. Hemophilia is a recessive sex-linked trait. Color blindness is recessive sex-linked trait. Muscular dystrophy is recessive, and Fragile X syndrome is dominant. These are just some examples. Now, then we come to, so the point of telling you all this was that when a gene lies in the sex chromosome, the rules are exactly the same. You can still apply the Punnett square, but the results will look different. And as long as you understand the basic construction of Punnett squares, you should be able to answer these kind of questions. Now, then we come to chromosome disorders, okay? So, there are 4 different kinds of disorders that happen in chromosomes. There is chromosomal deletion, there is chromosomal inversion, there is chromosomal translocation, and there is chromosomal non-disjunction, okay? And why does this happen? It happens because you have to go back to meiosis. In meiosis, you have these threads forming and separating the chromosomes out, right? In the first phase, it separates the two homologous chromosomes, and then it separates the chromatids, right? That's the way meiosis works. Now, and during that process, we also learned how the recombination worked, right?
Part of it breaks off.
The part of it breaks off, and that just, if you put two chromatids very close to each other, this just randomly sometimes happens. Now, in the process of this pulling with strings, if you think about 2 objects and you're trying to pull it,
Well, why does it happen? Like, if there's 2 next to each other, is it some kind of force pulling them apart, or like?
it's not force pulling them apart. The structure of the chromosome, which is comprised of DNA,
Yeah.
they can randomly kind of swap places.
Mhm.
It's
So there's just nothing happening that makes it swap places?
it's just because of the chemistry of this molecule, and I really can't go much deeper than that. It's because of the way the bonding happens in the molecule. This can happen. However, the other things that can happen is when you are pulling something out using some strings, sometimes if things are perfect, it just pulls apart nicely, right? But sometimes you can just pull it, let's say the string on this side is a little bit weaker, you might just pull the whole thing, right? That's one possibility. You might pull half of it. You might pull a different part of it. And these are all the result of that, these disorders. So this is not normal. So let's look at single chromosome disorder. So here what happens is, this part of the chromosome just got deleted. That's a deletion. So the gene just got lost. So
How did it get deleted?
how did it get deleted?
Yes.
During the process of meiosis, when it was getting pulled out, just a segment broke out, and that's it. And then the lower segment just joined the upper segment. So
I think it
That's-
happened when you have crossover. Remember during the line-up of the chromosome in the
Mhm.
middle of the spindle, and there are hybridization at the end of the chromosome, and crossover happened, so you lost part of it. Sometimes crossover happen like an A shape, okay, or a loop shape, and then you just delete the one part. You make a loop and then you lost part. When the chromosome cross over and make a loop, it's either exchange or it just lost in the process.
And then this is another example where it duplicates itself. Okay? So
And how does it duplicate itself?
again, the same thing.
It's by the misreading. Sometimes when the chromosome replicating, there's a promoter and there is the starting points. When the starting points got messed up, and it just continued to read and then read it twice.
So and then this is the inversion where
It just flips.
it just flips. But if it flips, it becomes a completely different gene. It's no longer the same because you're going the other
Code
direction.
changed, yeah.
Cool.
And then two chromosome disorder, this is called translocation. This is between two different chromosomes. Things can get deleted or added or swapped. Okay? And all these result in
Bad things.
bad things. Usually, most of the time, these are all bad things. Now,
Down syndrome.
once in a while, very rarely, it might result in a good thing. And that is the basis of evolution. That is how species evolve and become something else. But most of the time, these are bad.
things.
So, example, Down syndrome. In Down syndrome, it can happen either due to translocation between the chromosome number 14 and 21, and in another case, it could happen because of trisomy. And in trisomy, what happens is the entire chromosome just comes to one side. Remember in meiosis, you're supposed to get one chromosome. But what happens if both your chromosomes just move to one gamete? Then you just get an extra chromosome, right? And in this particular case, it's the 21st chromosome that you just got the extra one.
So what happens when?
What happens?
Yeah.
Bad things. This is what happens. You die in your maximum by your 30s, you're mentally retarded. I mean, you may have seen some of these unfortunate people in say Costco. I've seen people in Costco who have this disorder, so yes.
The class has like a rainbow sign. That's like the mentally disabled kids, and I went in there once. It's really sad
Hmm.
because I was helping my teacher because
Right.
I have a microphone condition.
Right, right. So if you think about it, it is just a matter of chance. During meiosis, in one of the parents, one particular gamete just happened to have this problem. And then, yeah,
What are like examples of like the mutation that actually benefit your system?
That's a good question. Is there an example of a mutation that benefits?
Like for the horses, they used to have like a toe, so then like one horse might have like lost the toe. And
Hmm.
so that might be an example.
What?
Nancy, do you know of an example of?
A lot of mutation actually are benefits, otherwise we wouldn't be here.
Yeah.
Okay, everything you are here is because the mutation from bad to good. So they don't really talk about mutation to, from bad to good because we all survive. Only they talk about the mutation that lead to disease or lead to sickness or lead to death. That is, people are studying. So, all the good mutation is what happened to you right now.
Mm.
So that's evolution. So the good traits, the good mutation all survive. So that's where we are right now.
So
From the AIDS.
here, this is an example of monosomy. Yes.
What's menstruation?
They won't ask you. So in this case, there is only, this is chromosome number, this is X. See, you don't see another X, right? Normally, in a female, there should be two X's, right? Here, one X is just missing. And so this is a problem. So this is something that in Science Olympiad, they will ask you to say what is wrong with the karyotype. And you'll have to look at it and say, chromosomes always come in pairs, and if something is missing, then you know it's monosomy. And if there is more than two, then you know it's trisomy, right?
Could there be
That's-
like more than 3?
Yeah, there is. I think there is. You could have even more than 3. So this is, here you have 47 chromosome. You have a XXY. So if you see, this is sort of like,
Super female.
no, there is a Y. So there is a bit of male, but the full female. So
So would that be male or female?
It's
neither.
it is sort of,
It's going to be a male, but
Male.
has a lot of female
Yeah.
features in the body. Are they going to expect to remember the name of the syndrome?
no. I'm just
Do
showing
they
them.
need to match the syndrome and the disorder?
No.
The karyotype,
no,
I think.
No,
they
they
might
don't.
give.
They don't ask about it, but I'm just showing them all the different kinds of things that can happen. So here, for example, this is not either an extra chromosome or a lack of chromosome.
Deletion.
But if you see here, one part of the chromosome is just missing, right? Number 5, deletion of the lower arm. And this leads to another problem. And here the upper part is missing, and again another problem. It leads to deletion of the upper arm. So tragic.
What does deletion? Oh, so there's no upper arm.
Yeah.
So like this part is just here.
Yeah,
Oh.
it's pretty sad. Again, here, deletion of the lower arm, so they don't really have legs. Anyway, I'm not going to go over. Yeah, this is, you can see,
Oh.
yeah.
Triple X chromosomes. There's such.
Yes.
What's a triple X chromosome? 3X is
3 X's
Three
instead of
X's.
2 X's, which you normally have in a female.
How would that even? Oh.
Yeah,
Wait, does all of these happen like by luck, or is
this
The
is all
chance.
by luck. It's bad luck actually.
Some of them aging, we've seen. Age matters, some factors, yeah.
It's all by science.
Yeah. So the
But
older you grow,
it can be
Chromosomes.
inherited.
And inherit.
yeah,
It's
yeah.
by luck, but then it can be passed down to the next generation.
Yes, yes.
It's like sex-linked dominant, recessive.
This is another example, the Duchenne muscular dystrophy. Here your muscles just keep becoming weaker and weaker, and eventually individual doesn't survive usually beyond like the 20s or 30s.
does it have to be a boy?
It's
Sex-linked
usually
will
in
be
boys.
boy, yeah.
It's usually in boys.
Because it's
Now-
Cystic fibrosis is common.
No. Now, not everything is in the sex chromosome. It can also happen in the other chromosomes, as you saw. And sometimes you'll see this term called autosomal dominant. Okay? Autosomal means it's in the non-sex part of the
The
chromosome.
body chromosome.
And it's a gene that usually causes, it's a version of the gene, like the lethal allele. This is a gene that causes a problem. In this case, it's Huntington disease. And it is not recessive. This is dominant, which means even if you have one version of it, it's going to affect you. And in case of Huntington disease, you'll only know it by the time you are in your 30s. That's when it shows up. So you could have your children, so you could have passed on this disease. So your children, if they happen to get that copy of your chromosome, they will also have it. And it's dominant, which means if there is one copy of it, you have it. And so there is the study of these chromosomes is called karyology. And what you saw earlier, the chromosomes all laid out in the picture, these are called ideogram. And it's a diagrammatic representation of the chromosome set of the species, and you can use this karyotype to see if there are any problems compared to the normal. So usual questions that they may ask: is this a male or a female? Is there any problem with this person? And if there is a defect in a particular gene, what will happen to their children? That's the kind of questions that they'll ask you. But it's important to understand what the like how to read this karyotype, and that's why I have this. So this is a very typical thing that you'll see in your yes.
Sorry. Karyology is the study of like the disorders in chromosomes?
No. Karyology is the study of Not study, it's actually a representation of the chromosomes in this particular form. Because normally in a cell, the chromosomes are not laid out like this, right, in a nice square shape. They are kind of floating around everywhere. So this is putting it all, yeah?
For the, I didn't want to interrupt, but for the 23rd chromosome,
Mhm.
it says X. Is there a reason for that?
Both of them are X's.
Oh.
So this is a female.
Oh,
This is
so it's
exactly.
saying, oh, it's saying that's the sex chromosome.
Yes, and it's showing two of the same size, which means it's an X and an X. And this is a typical thing that they will give you. They'll say, is this a male or a female? Okay?
Okay.
It's what?
She was talking.
Oh, yeah. Sorry, Nandini.
No, I
You were-
mean, no, you were talking, you were answering the question.
Oh,
I'm now
wait, so
confused.
karyology is the study of chromosomes, right?
Karyology is not the study of chromosomes, it's the representation of chromosomes in this form.
Then what's a karyotype?
Karyotype is, each of these are basically, okay. Karyotype is this picture, and karyology is the
You
study that leads to this kind of a picture. Yeah.
A question about the sex traits.
Mhm.
Why do so many traits show up, like perhaps like different types of cancers? Why do they show up in like later ages? And why do they not show up earlier?
Well, cancer is not always,
Well, those that are sex linked.
cancers may or may not be sex linked. Cancer usually happens because of some mutation that happens during your mitosis. It's your basic set of genes determines the chances of that happening, but a lot of the times it's also because of the environment. So, just, I mean, there is nothing in your basic set of genes that will say that you will have cancer for sure. Cancer is not like a Huntington disease where there is a defective gene and then something bad will happen.
And that's like why there's like cancerous like sprays and stuff like that. Like there were some
Yeah,
like, bug
exactly.
sprays, like,
So
cancerous.
environment plays a big role in cancer, and during mitosis, again, if you think about what happens during mitosis, your cells are just dividing and making copies of each other. Now, suppose there is a mutation that happens there, and that happens to kind of make a gene or a set of genes go silent, or
What do you mean
or-
by go silent?
go silent means they
Like
stop doing their function. So either a particular protein is not going to be produced, or a protein is going to be produced in excess. And either of these cases can just lead to the cells. So some of these proteins regulate like the cell cycle. Because if you think about it, if cells just keep doing mitosis all the time, right? You just keep growing infinitely. So it can't happen. It makes sense, right? So there must be some mechanism by which there are breaks, by which you say, okay, you should do it at a particular rate, which should sort of match the rate at which cells are dying, right? And all this is very, very complex protein pathways that are there. But if something goes wrong there and you suddenly stop getting the signal that stop, you just keep going. And there's nothing to stop you from doing your mitosis, multiplying.
So then what happens at that point in time?
Tumors.
Well, you have a tumor which if you're lucky and detect it early, then you can probably remove it. And then all those cells which were in that mode just completely got removed. And then they do all this radiation or chemotherapy that that kill those cells, and if you're lucky, then you survive, and if you're not lucky, then that's it, right? I mean, that spreads all over the body, and that cell, that tumor just shows up in different places, and eventually you just die.
So, but if you keep growing, then won't you just become very like tall or something like that?
Well, unfortunately, before that happens, that's a nice kind of growth, right? Before that happens, it'll probably cut off the blood supply in your brain, or cut off the blood supply in some place which will kill you. Okay. So these diseases are not directly related to cancer. This is problems that happen even before cancer happens. This is at the stage of meiosis. Okay. So the other thing to note here is not all chromosomes are the same size. Some chromosomes are bigger than others, and if a chromosome is bigger, generally it has more genes in it. So more genes in it, more information in it. Okay. So in human beings, for example, the first 3 chromosomes are the biggest, and then they progressively keep getting smaller.
But the 23rd one is not that small.
Well, the X one is special. That's kind of the sex chromosome, but and when the way they do this is by collecting a cell and putting a dye during the metaphase. That's when the chromosomes coil up like this nicely, and there are different dyes that they give by which they can see this banding structure, and this is how they determine like where the genes are located physically in the microscope, and also the position of the centromere is different between the different chromosome pairs. So you remember the metacentric, acrocentric, telocentric. Different chromosomes have different such properties, and this whole picture is called the karyotype of the organism. For example, this is the karyotype of a human being. So, what is this?
A male.
A male.
karyotype.
Good. Yeah, this is again another picture.
Male.
Yeah. And as you remember, this is human beings, and human beings N is 23. There are 23 pairs. So this is a diploid organism. So the chromosomes always come in pairs. You'll hear diploid and you'll hear haploid. Haploid means there's only one, and that's only for the gametes. There are other, like, there are more than 2, but
How does
that's...
the diploid split?
Huh?
How do the diploid
That's meiosis.
That's meiosis, right?
oh,
You had these two,
yeah.
and then they
you
just
split,
assorted.
Yeah,
And,
so yeah.
yeah. And by looking at these chromosomes, today you can actually sequence the chromosomes. You can know exactly what the chemical composition is. In the past, people used to look under the microscope, and by putting dyes in the chromosome, they could actually see where the genes are located physically. So this is the different places from where you can draw your cells, and then you can do the karyotype. So the chromosomes are classified according to the site at which they have the centromeres. We already discuss that, and according to size and then different bandings. Anyway, this is in human beings. The group A are the largest, and they are metacentric and submetacentric. So they will have their centromeres somewhere in the middle, and then 4 and 5 will be submetacentric. So it will be somewhere here, and then there is medium submetacentric. It's probably not very important, but when they show a human karyotype, which is what they will do in Science Olympiad questions, you should have an idea of what is like. If something is deleted, you should be able to pick it up from the picture. Okay. Okay, so that's pretty much. So this just says how this karyotype is done. You take the cell, you incubate it in a culture medium for a few days, and then you add some chemical that stops the mitosis in the metaphase, and then they are put in a. So during the metaphase, what happens is the chromosomes coil up and come near the equator. So now you take those and you stain it and put it under the microscope and get this picture. Okay? Any questions? If there are none, I will go to pedigree analysis. So, pedigree analysis, what it is today, I mean the last 10 or 20 years, a lot of DNA sequencing has happened, and people can actually know exactly what gene they have in their chromosome, at least for the known genes. But in the past, people were aware of chromosomes and genes for the last 70, 80 years. And you can go to a genetic counselor, even today you can go to a genetic counselor and tell the history of your family and say, okay, so and so in my family had this particular disease. What are the chances that you need to leave?
Yeah.
Yes.
Sorry.
No
I'll
problem.
leave the video on the.
Yeah, thanks.
Thank you.
I should be done in another 15 or 20 minutes.
Okay.
Okay. I'm not going to draw anything in the board, so you should be able to
It's
watch
just,
the.
okay,
Yeah.
on the PowerPoint.
Everything is on PowerPoint today. Yeah.
Thank you.
Thank you. Bye. So you go to a genetic counselor and you tell him about your family history, and usually, I mean, from research, people knew whether a particular disease was linked to a particular gene, and whether the gene was a sex-linked gene or not sex-linked gene. And based on this information, you can draw a chart of that particular disease, let's say. And that's called pedigree analysis. And from that, you can draw conclusions about what are the chances of somebody having the disease or passing that disease to the next generation. And that's really what pedigree analysis is. Now, even though it follows Mendelian ratios, usually people have only a few children, right? Not like hundreds of children. So Mendelian statistics holds good when you have a very large samples, because that's when the law of probability actually converges. If you have one or two children it might so happen that none of them have the problem or both of them have the problem. And it doesn't really follow the ratios that you'll see. That would have happened if you had a larger sample size. Okay. Okay, so these are the symbols that you'll see for your oops.
You can slide the bar.
Huh?
You know, there's this bar for zoom out.
For some reason effect slider should work, but for some reason
No, I know, but I think it's enable editing and then you can do it.
maybe you're right.
Genetics.
I'm just it's just telling me that oh something bad okay. Maybe okay, good. Okay, so most common symbols. So the square is for a male, the round is for a female. That's and a black is that particular disease or disorder that you're studying. If it shows up, that means the phenotype actually shows up, then it's an affected male. And same thing for female. And then other common symbols. This is important, by the way. You have to remember this. They will ask questions. Okay, so this says that a male and a female, they are mating, and then they have children. Here you have two parents who don't have a disease or that particular disorder, produce two children who don't have that disorder, and one is a boy and one is a girl, right? And then here, for example, this one is heterozygotes. That means they have one dominant and one recessive. And autosomal recessive, which means it is not in the sex chromosome, it's in the autosomal chromosome, and the disorder is recessive, which means it's not going to show up. So a person like this is a carrier of that particular disorder, but it's not going to show up for them. They will not suffer from that disease or disorder. Yes?
On the bottom on the left it says sex unspecified.
Yeah, I don't know what that is.
It's either mono.
Though I haven't actually seen a question in Science Olympiad which has this symbol.
Is it because the trisomal?
Yeah,
It's the black one that looks like a bomb on the other side.
It's a stillbirth. It's a stillbirth. That's why they
It's
do.
abortion or stillbirth.
He's
It's
so
like a
Right.
bomb.
My mom was like a bomb.
Looks like a bomb.
Okay, the other thing. Now you have to remember this, by the way. A circle with a little dot in it. It's also recessive, but it's sex linked. So it's not on the autosomal. So that's the way you distinguish between the two. This means he's dead. He was not a carrier, but he's dead. He or she is. In this case, he is dead. And this is the sign for monozygotic identical twins. And this is the sign for dizygotic non-identical twins. And you know the difference between the two, right? Identical twins and non-identical twins?
Yeah.
You know, okay, good. Anybody doesn't know?
I would love to have that.
Yes, we would.
And here, for example, you see you have an unaffected male marrying a affected female, and they have 3 children. One, and they show the generation through these Roman letters. They have 3 children, 2 boys and a girl, and the boy gets it, right? So
And what's propositus?
propositus.
The one above.
Propositus, the propositus is the child to, propositus is the person for whom you are seeking,
Oh,
like,
you're asking about.
you're asking about, yeah. Usually you go to the genetic counselor, and after you give the history, he or she will come up with this chart. And in this particular case, this is the person
Inquiring.
you're inquiring about. So here, cystic fibrosis. You have a male and a female. They have 1, 2, 3. Okay, sorry. They have 1, 2, and 3 children, right? And this is marriage. This is mating. So they have 3 children, 1 boy and 2 girls, and the boy has cystic fibrosis. Now, usually if you have cystic fibrosis, maybe he didn't marry, but the girl's married.
What is cystic fibrosis?
It's a disorder where you have your lungs doesn't have the ability to drain the mucus, so you just accumulate mucus and
You cough.
and
Infections.
yeah.
Like
you
repeated
eventually die
infections.
because of
Do people eventually? I think they
Depends.
will die
Of
by
respiratory
40s.
infections.
By 40s they will die, but now there are so good treatment that they survive till their 40s. But yes, they will die earlier than normal people. And
Because of the infections.
yeah, you will still have to have someone who goes and removes the cuff from their chest physically. They have to pump their chest. So they probably have to go to hospitals, or somebody has to come to their house to do this. So this is how you read a chart, okay? And you tell the history, and you say, okay, my grandparents had 2 daughters and one son, and my uncle had this cystic fibrosis, but my mom and my aunt, they were normal. My aunt's family is completely normal, but I have I have 4 siblings, and I have cystic fibrosis, but my 2 sisters and 1 brother is fine. That's what you go and tell him, and then this chart will be drawn. Okay. So now you guys can read a chart, right, like this.
Yeah.
I know I'm probably going a little bit over time, but since I'm here, why don't I just finish this off? So I just described. So this is called pedigree analysis, okay, or a
So
pedigree
just
chart.
making the chart is called pedigree analysis?
No.
This is called the pedigree chart, and from looking at this chart and drawing conclusion is called pedigree analysis. So the conclusion is, okay, my mom had this or my uncle had this, what are the chances that my children will have it? And once you draw this chart, you can sort of make a prediction. So in this case, you're assuming that this is a recessive gene. So obviously the gene must have been there. The reason why it's white is it didn't show up, right? The gene must have been there in one of the parents, and in this case, this doesn't look like a sex-linked disorder
Because
Be-
male and female.
because
have
both males and females are having it. And so this particular person must have had this AA copy. So we don't know exactly how it happened. Maybe this was capital A, capital small a, and this was capital A, small a. Actually, it had to be that, and then this person got the small a. Now, here, this must have been a capital A, small a, and she happened to marry someone who also happened to be a capital A, small a, and so, so one of them got it. So this is how you draw the conclusions, right? So this is most likely a autosomal recessive gene that is causing this problem, and that's what cystic fibrosis is. What's the time?
7:30.
7:28 to stop.
7:28. Is it okay if I go on for a few more minutes? Huh?
No.
I don't want to drain you guys out,
Yeah.
guys and girls out. But we are almost done with this, so why don't I just finish this off? So here, what are the genotypes or possible genotypes for each individual in this family? Use the letter B to represent the dominant alleles and small b to represent the recessive alleles. So again, this is showing up in both male and female. So most likely, not most likely,
It is also.
it is an autosomal recessive, and neither of the parents had it, but unfortunately, two of the children happen to have the small b, and so here are the different things that can happen. Autosomal recessive, you just saw the example of that. None of the parents will show the problem. Autosomal dominant, where one parent will show the problem, one or both parents can show the problem. But the problem is that by the time they show the problem, they already have had children. Okay. X-linked recessive, this means it's not in the autosomal chromosome, it's in the X chromosome, and it's recessive. If it's recessive, then if you're a girl, you will not see the problem because it's recessive. But if it goes into a boy, the Y cannot stop the recessive, so the boy will show the problem. Okay?
But it can also be in female if the both are recessive.
Yes, that is correct. If you have both copies of recessive, if you're homozygous, then it will show up. Actually, that is not true. Because, okay, this is another interesting thing. In females, you have two copies of X, and soon after, like mitosis, one of the copies actually just shrinks itself. So really, only one copy of the X is active.
So it shrinks itself.
Kind of,
Inactivated, it's
It
called
gets-
X chromosome inactivation.
yeah, it inactivates itself. Yeah, physically it shrinks itself. or
Basically, it
something.
remains the same, only thing
Oh,
it's silenced, all the
It's
genes
silenced,
are
all the
silenced.
genes are silenced. So if it's recessive, so this is the other, yeah, that's a good point. If it's recessive, it is not going to show up because the other X chromosome basically is having no effect. You have X-linked recessive, X-linked dominant, which means the allele is found on X. It can be both male or female, and hypophosphatemia is an example of that. I'm not exactly sure what happens in this. And then if it's Y-linked, it'll only show up in the boys, in males, and it'll only get passed from father side. It'll never impact a girl. Yeah.
Are there any examples for Y-linked?
Example of Y-linked. Nancy or Simpson, do you know?
Probably like infertility or something like that, because Y chromosome mainly codes for genes important in male characteristics, like production of sperm and other characteristics. So if those genes are, you know, defective, then you'll be sterile or something like that.
Okay, so example of autosomal recessive trait you already saw: cystic fibrosis, sickle cell anemia, PKU, Tay-Sachs disease. Autosomal dominant. In autosomal dominant, you can see
Look at the pedigree.
7 generations.
every pedigree will show this,
Five generations.
right? And that's because it's dominant. If you happen to get that copy from your parent, it'll just show up.
There's 5 generations.
There are 5 generations.
Roman numerals.
5 generations.
They have
Why
five
are there
children.
so few offspring in the 5th generation compared to the 4th?
Go and ask the
I have a
generation.
question. So in the
You
third,
guys notice things which I
We
never
don't
thought
notice.
about.
Why the fifth has less offspring?
It just shows that human beings nowadays have smaller families
Yeah.
than in the past. Like my grandparents had like 10 children, so out of that only
We have one now.
4 survived.
in the third one, in the third generation,
No, that's not what I
there's
meant.
that one family with a lot of children.
Mhm.
So in there, 2 of the children are with inter thing. So how?
Yeah, that's called like a consanguineous
Consanguineous marriage.
marriage.
It's within the
Your
family.
brother married the sister.
Fries. Double word.
Oh no.
Consanguineous.
Double.
Yes.
Why are there 3 question marks?
Why did I question mark?
Or why are there
In
question
the
marks?
5th.
Because they don't know what the sex of these children are. And
No.
Where's the diamond?
No, it says.
I see a circle.
It says circle.
They don't have any offspring, they don't know.
I think they didn't
No
match.
offspring.
No offspring.
No off or unidentified.
no, this
Oh,
shows
but they are diamonds. They are diamonds.
They are diamonds, right?
If you
Oh
look
yeah, they
closely,
are
they are
diamonds.
diamonds.
3
Then they why did they ask why they ask
Wait,
the question?
from the circle? How did why is there a line going down from the circle? Like just one thing. There's no line in between connecting to the diamond.
Yeah, this one,
This is a circle, then diamond.
They randomly became.
What?
Maybe they don't have any history, so they just put it.
Oh.
There is no record.
There is no history.
yeah,
So
No, there's no history.
To the
No history, no
Okay.
information.
no information.
Sometimes you can't give information of those siblings.
Okay, so you guys should be able to read this, right? Read this pedigree chart. And then they will ask questions like, what do you think? Is this a recessive-ish or a dominant? Is it sex-linked, not sex-linked? So sex-linked, if you have sex-linked, it'll show up, it'll only show up in one particular, either affect only males or females. So this is an autosomal dominant trait, dwarfism. Have any of you seen dwarfs? So, yeah, okay. So dwarfism is actually a dominant trait, which means if there is a parent who is a dwarf, there is a 50-50 chance that his children will be dwarfs, because it is dominant. Huntington's disease is also an example of autosomal dominant. So you can see it shows up and then it's affecting both males and females. In the X-linked recessive is, if it's X-linked recessive, it'll show up only in males, right? Because the recessive version in females is not going to show up. And males are more often affected. Okay. Hemophilia. This is a famous example of the royal family in Britain. This is Queen Victoria.
This
is the royal family of England.
9 generations.
9 generations.
And
You'll go home and study this.
people believe that this happened due to a mutation in Queen Victoria, because none of her ancestors showed this characteristic. And what happens in this particular case is you'll bleed to death, because the agent that does the clotting is
Blue blood, right?
whatever, it's absent. So Queen Victoria, you can see here, you can see these circles with dot inside. What does that mean?
The female
The
carriers.
carrier.
Carriers, it's sex-linked and it's recessive. Remember? If it was not that, if it was autosomal but carrier, how would it have shown?
Then I would have shown it.
half of it.
Half of it would be black.
Yes.
So this little circle in the middle is saying that it's sex-linked and it's also recessive. And see, it shows up only in men in the family, and it shows up in many men. And the reason for this is, in males, even though it's recessive, the Y chromosome doesn't provide any protection. So if they happen to get that particular version, that's it. Then bad luck. And then all the females are carriers, right? Okay, so this is X-linked recessive trait.
There's a light in this ear.
Oh, I thought his ear was.
Yeah, this is some. If you eat fava beans, then it basically is bad. Excellent dominant pedigrees. So you see, it's there in the male and it's dominant, and then it shows up in females because the female has to get the X copy from the father. There is no other choice, and all of them have it because it's dominant. Actually, that's interesting. This one doesn't have it.
It's not connected
That
to
one is
the,
not the,
it's not connected.
that one is not the,
It's not
it's
not
not
the
the
wife.
it's
It's not the wife because
It's the wife, it's not from the same.
Oh, I see.
It's a marriage.
Yeah. Okay. You're right. Yeah, you're right. So you can see, right? And you can read this chart and make these predictions, right? Dominant traits are rare in population. You know why, right? Because